Hello, everyone. We'll be discussing the historical figures and events in radiation oncology in this session. And our main aim is basically to understand how the field of radiation oncology developed over the years. And by the time we end this session, I hope you'll be able to recognize these three pictures that I have on the screen right now. Now, to begin with, the first person on the screen is none other than Röntgen. In 1895 in Germany, he discovered X-rays. He first took the X-ray of his wife's hand, Anna Bertha, and then later at a public lecture, he took an X-ray of his friend, Albert. This person, Antony Henry Becquerel, in 1896, gave the concept of natural radioactivity. Now, he said that there might be some elements that give off natural electromagnetic radiation. Now, X-rays had been around for like a year only, but people wanted to apply it into the medical field to treat tumors and other benign lesions. So this person, Emil Hermann Group, in 1896, he treated a woman with recurrent carcinoma breast with an X-ray tube. These people, Schiff and Freund, in 1896, treated pigmented nevus of this lady. This picture was taken in 1956, that's like almost 60 years later. The nevus is not there anymore, but she definitely has an adverse reaction, which is the kyphosis. This person, Victor, he used X-rays for palliation. He treated a patient of carcinoma stomach, and he reported almost an 80% decrease in the pain. I hope many of you people recognize these two people. This is Marie Curie and Pierre Curie. They are the discoverers of polonium and radium. And radium forms the basis of brachytherapy treatment for the next 80 years or so. In 1900, in Stockholm, Thor Stenback said that fractionated radiotherapy can be used to treat skin cancers. But the apparatus at that time which produced X-rays produced very low energy X-rays. They had very poor penetration and could treat only superficial and accessible tumors. Like these two people, Danlos, the Ehlers-Danlos syndrome person, and this person, Eugene, he's a physicist. They used radium tubes to treat skin lesions. In 1903, this person Cleves, he reported the first case of cervical cancer cured by X-rays. So see, we are treating skin lesions or accessible lesions like the uterine cervix. At this point of time, some changes also began to take place in the field of radiobiology, like Henke noted the radiosensitivity of lymphoid cells. By the way, in 1906, these two people, Bergioni and Tribandiu, they linked radiosensitivity to proliferation rate. That more the proliferation, more the radiosensitivity. In 1910, in US, this person Abbey first implanted radium tubes directly into sarcomas and carcinomas. So this was like the beginning of the interstitial brachytherapy. By this time, people began to notice the adverse effects of radiation. So in 1911, Jagic reported cases of leukemia in radiation workers. And further in the field of radiobiology in 1912, this person Schwartz, what he basically did, he would press the X-ray tubes into the skin. And he realized that when he pressed the X-ray tubes into the skin, there was less radiation dermatitis. So that means he basically postulated that when he presses it into the skin, he decreases the blood flow to the skin, empties out the capillaries and all. So maybe that decreased blood flow decreases radiation response. Now in 1913, William David Coolidge, he made the hot cathode X-ray tube. Now this invention basically revolutionized production of X-rays and actually gave us a step into producing the 200 kV X-rays. So the 200 kV X-ray tube came into being, the ortho voltage. It still could not treat very deep tumors, but it was better than the low energy X-rays we had been using up till now. Now, because deep tumors could not be treated, there was a scope for teleradium therapy for treatment of deep seated tumors. In 1922, the first case of lung fibrosis after radiotherapy was presented. And by the time 1923 came, we were able to make some isodose distribution diagrams to study how the dose deposited decreases with the distance that it traverses into the body. So, also in the field of radiobiology, Petri showed that there is a correlation between radiosensitivity and oxygen. So, there is development in all the fields together. Now, in 1927, Muller observed that X-rays causes mutations in Drosophila. Now, people wanted more of treatment and less of adverse effects. So, in his animal sterilization experiments, Claudius Rigaud in 1927 France realized that if he gives less amount of radiation every day to the scrotum of a ram, he was able to sterilize the ram without causing any skin lesions. So he realized that fractionation over a long period of time gave less adverse effects and gave a similar outcome. Similarly, in Paris, Henri Cotard, he found that in patients who were treated for a longer period of time, they had lesser radioepithelitis, that is, of the mucous membrane, and dermatitis of the skin. So he could see that the patients who were treated in 10 days had more of mucositis and dermatitis, while the patients who were treated for like 18 days with the same dose had less of both. So this was like the basis of why we give a fractionated radiotherapy today. Then this person, James Heyman, he developed a packing technique. This was basically a radium tube. How to pack this radium tube into the uterine cavity for a better treatment. So he developed this packing technique for brachytherapy. While in the field of radiobiology, Mottram showed that there is an importance of oxygen in the radiosensitivity. From 1937 to 1940, there was a rapid development in the microwave technology. Microwave had been around for the last 20 years or so, but we were not able to make full use of it. It was being used in radars and all, but these two things were developed at time the klystron and the magnetron. And in 1940, Donald William Kirst developed the betatrons. This person, Donald William. These three together formed the basis of the modern LINAC. So in 1943, this physicist Fry gave the first diagram of a zero point 0.5 MeV linear accelerator. Up till now, we were working with around 200 kV extra tubes. With this linear accelerator, we could produce high energy X-rays. But the concept was given in 1943. But the first LINAC could not be installed till the next 10 years. In 1948, this person Ellis, he noticed that the response was different depending on the number of fractions given and the time over which radiotherapy was given. So the work on fractionation was going on actively. From 1950 to 1955, cobalt 60 came into being. So radium teletherapy machines were being replaced by cobalt 60 machines. And Harold E. Johns in Canada, the first patient was treated with cobalt 60 in Ontario. This person, Harold E. Johns, he was actually a physicist and he was very, very active with establishment of the cobalt teletherapy machines because high energy linear accelerators were still not in being and low energy x-rays could treat only superficial tumors, so we needed something to treat deep seated tumors. So cobalt 60 at this point of time was so revolutionary that people thought that it might replace x-rays in radiation treatment. However, that did not happen. This is a very old cobalt therapy machine. It is called the Eldorado. You can see the Eldorado here as well. And this is also a telecobalt unit by the company Acme. Now, this person in 1953, Louis Harold Gray, he first carried out the quantitative studies on oxygen and radiation induced growth inhibition. Basically, the relationship of oxygen, blood supply, tumor reduction, it was going on, but there was no quantification in it. Nobody could actually study it up till now. But this person, Lewis Harold Gray, was the first person to do so. With the help of this person, Thomilson, he proposed that oxygen level decreases in the tumor mass with the distance from blood vessel. The more the tumor mass is away from the blood vessel, the less the oxygen. The less the oxygen, the less the radiosensitivity. And interestingly, this is the person after which the SI unit of absorbed dose has been named, Gray. In 1956, these two people, Puck and Marcus, who were quite good friends with Gray, developed a cell survival assay. Up till now, we knew that the tumor is decreasing in mass or number, but the cells on a cellular level, we had not been able to we had not been able to study it. So they developed an assay. Basically, they plated HeLa cell colonies, and then they discovered the number of colonies that were formed after irradiation with X-rays. So they were the first to do so in mammalian cells. And now in 1957, that's like almost 10 years, the first linear accelerator was installed in Stanford, US, which treated this 2-year-old boy for an eye tumor. In 1959, another cell assay was discussed by Herbert and Wilson, and they produced the first cell survival curve. That is, they plotted the logarithmic rate of survival of the cells against the dose of radiation given. And you can see, even at this point of time, they're using the classical RAD unit of radiation. In 1960s, new artificial radionuclides were developed for brachytherapy, like the cesium-137 and the iridium-192. And iridium-192 is still one of the most important radioisotope being used. Now, this person, Mortimer M. Elkind. In 1965, he discovered sublethal damage repair, and he also linked it to dose fractionation. Sublethal damage repair is the most important concept that one comes across in radiobiology. It is the basis of both the repair of normal cells and the survival of tumor cells. And this shoulder that we see here is basically the sublethal damage repair. He also linked to fractionation in his further studies. You will come across this concept everywhere in radiobiology and this diagram as well. In 1966, high-dose radiotherapies units have been invented, and afterloading mechanisms had come into being. Up till now, the radium needles were being hot loaded. They were being handled by personnels in presence of all the radioactivity, and when they were being inserted, very risky business. But with the advent of afterloading mechanisms, high dose brachytherapy could be given. High dose rate brachytherapy could be given with an afterloading technique. You don't need to be in the room when the patient was being loaded with the radioisotope. Also, in 1966, hyperbaric oxygen therapy was being applied to a radiotherapy patient. Hyperbaric oxygen therapy had been for around like 50, 60 years up till now, but it had not been used for radiation-related adverse effects. This was the first time in 1966 that it was used in Italy. Also, in 1966, the TCD50 assay was developed. It is another assay for um, cell survival, but for multi-hit response law. In 1967, Withers quantified the number of stem cells and survival of normal cells, and hypersensitivity was also seen in ataxia telangiectasia patients. Now, in 1968, Sweden, something quite revolutionary happened. Lars Lexell and his physicist developed stereotactic radiosurgery. This thing here you see is a gamma knife, and this was the advent of stereotaxy. In 1969, another very interesting thing happened. G.W. Branson, he developed the LQ model, the tumor growth delay assay, which is like the most acceptable model for the basis of radiation and how it kills cell and how it works. It is the most acceptable model at this point of time. Now, in 1970s, we all know that the revolution in the computer technology came. It improved not only our treatment planning, it also got integrated with CTs and MRIs and how we image the body today. Now, this person, Hounsfield, he was behind development of the CT scan, while these two people, Paul Christian and Peter Mansfield, this person Mansfield, with his first prototype of an MRI, and he imaged the first body part. It was an arm of a human. Now, in 1972, the concept of apoptosis and traumatic cell death, the differentiation between the two was being given, which further later developed into a very different field of oncology. In 1975, the PET scan was developed, but it was only a PET. This is the first prototype that you can see here. It's like not until the next 10 years, 15 years maybe, that we were able to integrate it with a CT scan. In the 1980s, the first clinical trials of combination of chemotherapy and radiation was started. And also, the establishment of proton therapy happened. Proton had been around since the 1960s, but it was quite experimental. It was there in only two or three centers in the world, but in the 1980s, proton therapy saw a boom, and the number of centers increased, especially in the US. 1980s was also the time of development of the multi-leaf collimators, which was developed by a number of companies, and this basically revolutionized our technique of treatment delivery. It led to the development of the three-dimensional radiation planning and the IMRT. Three-dimensional radiation planning was possible because of integration of CT scan into our imaging, while IMRT was possible because of the MLCs. This was also the time of development of bioreductive drugs. It was now in 1991 that this person, Railman, first suggested the combination of PET scan and CT scan. And these two people, Ronald Nutt and David Townsend, were able to do so by 1998. It was first installed in the University of Pittsburgh, and the first commercial system was available in 2001. So, this kind of brings us to what we have in contemporary radiation therapy. We have better immobilization devices like this breastport. We have better radiotherapy source like this iridium radiotherapy source. For brachytherapy, we have good afterloading mechanisms for high dose radiotherapy. And we have the modern linear accelerators. We even have the cyberknife, where the linear accelerator is mounted on a robotic arm. And we have the latest model of the gamma knife. So, this is basically a historical overview. And if you have more queries or any people that you want to discuss and add, please feel free to comment. And the treatment planning system also changed. We were initially not able to visualize how the dose is going into the body. Later, those isodose distribution curves came into being. So we were able to make an approximation. But today, with the computer algorithms, we are actually able to visualize how the dose might be overlying in the body. We are able to see the tumor in the PET, the CT scan, and then able to overlay the dose that is being given. With IMRT, we are able to better spare our critical organs like this spinal cord over here, the parotid glands, and even for brachytherapy, we are able to better visualize our insertion and how the dose might be given around the source. So I'll be more than happy if you put up more people in the comment and please share. Thank you.